Hi, everyone. Welcome to the fourth webinar in our six-part series on alternative approaches for acute inhalation toxicity to address global regulatory and non-regulatory data requirements. My name is Amy Klippinger. I'm the Associate Director of the PETA International Science Consortium, and will be co-moderating today's webinar with Dr. David Allen, who works as a contractor supporting the U.S. NTP Interagency Center for the Evaluation of Alternative Toxicological Methods, also known as NICETM. Thank you all for joining today's webinar. And before I introduce our speaker, I'll briefly run through a few logistics. So as I mentioned, this is the fourth webinar in our six-part series. If you missed the previous webinars, you can find the recordings posted online at the link on your screen. Today's webinar will focus on using the GHS additivity approach to classify mixtures based on ingredient toxicity. The slide from today's webinar and a recording will also be posted online within a couple days, again at the link on the screen. Everyone is muted now. If you have a technical question for me anytime during the presentation, you can use the question box, which is located in the panel on the right-hand side of your screen. If you have a question for our speaker, you can type it in that same question box, and David will ask your questions to our speaker during the Q&A period after his talk. So please feel free to send your questions in anytime during the webinar. I also want to point out that this webinar series is being organized with input from multiple organizations, which are shown here. And I'd like to thank each of these groups for their efforts in this series. And lastly, before I introduce today's speaker, I want to mention the other webinars coming up in the series. The next webinar on adverse outcome pathways will take place on July 12th. Followed by our final webinar, which will be held on September 8th. You can find more details about these webinars online, again, at the two links shown on the screen. Please note that if you're registered for this webinar, you're automatically registered for all 6 webinars in the series. But if you like to use Outlook calendar reminders, you have to add each webinar to your Outlook calendar individually and manually. GoToWebinar doesn't automatically transfer these to your calendar. So please remember to get these last 2 webinars on your calendar if you haven't already. Okay, so our first speaker today, or our only speaker today, is Dr. Marco Crivaro. Marco has more than 10 years of experience in safety evaluation for regulated industries. In his current role as regulatory toxicologist and human health risk assessor at Dow AgroSciences in the UK, he's a subject matter expert for regulatory and predictive toxicology of agrochemicals, from early transition from discovery to the post-approval phase. This includes responsibilities as human health risk assessor from both a dietary and non-dietary perspective. In addition, he is a European focal point for various aspects of alternatives to animal testing and 3R strategies. Previously, he worked as a toxicologist at Research Toxicology Center in Italy, developing knowledge of pharma, biopharma, agrochemical, and chemical requirements. During his academic experience, he developed a strong background in molecular and cellular biology, particularly in the exciting field of cell death physiopathology and its role in molecular oncogenesis, cancer therapy, and potentially cancer gene therapy. And with that, I will turn it over to you, Marco.
Thanks, Amy. I hope you can see my slides.
Yes, we can.
Yeah, perfect. Okay. Hi, everyone. I would like to give you a flavor of some of the activities we have done at Dow AgroSciences and presenting on the GHS additivity approach as a very useful tool to classify mixtures, and with particularly with a focus on acute inhalation toxicity. This presentation will be divided in 2 parts. In the first part, I will give you a little bit of background on the acute inhalation and the theory of additivity and how these have been implemented in the GHS in terms of theoretical calculation approaches that can be used to predict acute inhalation toxicity. And then I will go through some examples, and the main example I will describe is the agrochemical formulation example, where we have done a retrospective analysis, which we are publishing, on the power of this additivity formula for prediction of inhalation toxicity for agrochemical mixtures. So exposure to airborne particles may be found in some of the industrial products. With products, generally we intend mixtures which are composed by different ingredients. And this is particularly true when these mixtures are applied as a spray or sold to be used as a spray and or as powder or dust dispersion. The industrial products that have this kind of application spread in all type of industries. So we have industrial chemicals, agrochemicals, beauty care or house care, and then products, and then medical application, and also other fields. When we register a mixture, and this is different in different sectors, we may be required to perform inhalation toxicity testing. As I said, the requirements differ globally, and the differences are driven by geography and definitely by industrial sector. In some geographies, we have standard requirements, so we may be required simply to perform an acute inhalation testing in vivo or repeated dose inhalation testing, and there are no possible derogation or waiving criteria. But in other geographies, likely requirements are conditional, and those are generally determined by exposure scenarios. So various elements are taken into account. Obviously, maybe the order is not really right on this slide, but physicochemical properties are very important, like volatility, particle size, the dilution. If in the use pattern you expect to have the product diluted, and then obviously if there are information on the intrinsic properties of your mixtures, like severe effect, irritant effect, corrosive effect, those can be taken into account. And all this information will feed into essentially a weight of evidence, and you will determine if a study is necessary or not. For some of these sectors, data may be used for classification and labeling purposes, and in particular data on acute inhalation toxicity. Now, when we think to the acute inhalation, well, in general, to the toxicity, the inhalation toxicity of mixtures, it's important to consider how the various components of the mixtures are acting into this scenario and this exposure route. Often there is little information on a toxicity mechanism for each of the mixture components. So during time, there was development of different pragmatic approaches that can be used for determining mixture toxicity. We have very complex methods now, but if we go back in the history and we see at the very beginning, a theory of additivity was essentially formulated based on mathematical models. So essentially, Finlay in 1954 produced a mathematical model for mixture toxicity prediction based on additivity assumptions. And this theory was also verified in the following years, and you see here some references, essentially confirming that using this theoretical model for industrial mixtures, the predictions were correct when these data were compared to existing available in vivo studies. In addition, always from a mathematical perspective, it was estimated that approximately 5 combinations have less or greater than additive effect. So essentially, For acute toxicity, the additivity can be assumed unless there is evidence that a synergistic effect can be observed. Now, in more recent years, different literature has been produced for toxicity occurring at low doses. So at doses lower than the NOAELs, additivity may overpredict essentially toxicity of the mixture. And this is likely to reflect the differences in the mode of action of the various components of a mixture that can contribute differently or independently to the overall toxicity of the mixture. Now, this theory of additivity was brought into the GHS and also very early in some other regulations. So the GHS is the Globally Harmonized System for Classification and Labeling. But also other systems like the DPD in Europe, the Directive for Classification of Preparations, which is a 1967 regulation, then further developed during the second half of the last century, implemented this method for the prediction of toxicity of mixtures. So this method is a calculation method, so it's a computational method, and uses composition information, so your recipe essentially, and the toxicity of each single component as a starting point to calculate the toxicity of the mixture. The prediction of the systemic toxicity is prediction of acute systemic toxicity, so it's not applicable to repeated dose toxicity. And the prediction is actually occurs in terms of toxicity classes. So it actually gives you an endpoint value, but the final goal is to put your mixture into a toxicity class rather than predicting a single specific endpoint. This method is usable as a standalone non-animal replacement method in some of the geographies. And examples are given here. Obviously, GHS implementing geographies will accept this method. The EU CLP will accept this method, and some specific sector-specific regulation will accept this method. For example, in New Zealand and Australia, the regulation on agrochemical formulation accepts the use of this method. This method is also integrated in some other horizontal regulations, such as the transport regulation coming from the United Nations. So this, in case you do not have a study on a mixture, you can predict acute toxicity using this calculation method. And this information can be used to determine the packaging or the labeling of the compound. A very important point is that this method has a minimal cost and effort and doesn't use any animal. Now, let's go into the nitty-gritty of the formula, which is actually pretty simple, and you can see it here. This comes from the CLP. I wasn't able to cut and paste from the GHS because it's secured, but this is exactly the same. So essentially, you consider the concentration of each of your ingredients, and then you consider the ATE, acute toxicity estimate, for each of your ingredients. And you sum them as a function of their concentration. Now, where we have to discuss 2 points here. One is the source of information. So you will have your components, and you can gather information on the acute systemic toxicity from the MSDS or from available databases which have regulatory acceptance, like the ECHA classification and labeling inventory, and through the ACTOR EPA ACTOR website, you can arrive to solid information. Now, you can use the LD50 or the LC50 if this value is available for each of your components. But sometimes you will find that a certain component is classified on the basis of a range test, for example, an acute toxicity class test method, for example. So you will have an LD50 or an LC50 which is estimated to be in a certain range, or you will find that the LD50 or LC50 or the classification category, the LD50 or LC50 or the range is not available, but you will find that this component is a GHS category 3. So you will come back to the fact that is the LD50 or LC50 are in a range. So as you see on the table on the right side of the slide, let's take the oral case, for example. If you have a certain component which is a category 4 or essentially has an LD50 between 300 and 2000 milligrams per kilo body weight, you will assume that the LD50 is 500 as indicated in the column in the far right. So if you have an LD50 or LC50, you will use that value for that component. If you do not have that and you have a range or a category, you will use an acute toxicity estimate, which is what is displayed in the column on the right. Now let's go through some examples. First example, very easy. An oral LD50, there are no classified substances in the ingredient list. So essentially this method is not applicable because you cannot, you would expect essentially no toxicity and you would apply no classification. Second example, you have one component which is classified. This is a dermal LD50 example. You have one coformulant. Say that this coformulant Y is contained at 20% in your mixture, and your coformulant Y is containing different ingredients, one of which, benzene sulfonic acid, is contained at 4.5 something percent and has an LD50, dermal LD50 of 1100. You will calculate using the formula on the top of the slide the concentration of, you will normalize concentration with the estimated with the actual LD50, and you will calculate in percentage what is the LD50 of the final mixture. As you see in this case, you have a number which is 23,800 something milligram per kilo body weight, and this is obviously a mixture that is not going to be classified. So if you have a single component, you will take the concentration of the component. But if you have a single component which is a mixture, then you will take for the element of the mixture the concentration of that element, and then you will use the LD50 for the calculation. This is the third case is a case of an oral LD50, and that contains two components classified. Fairly easy. The components are classified, and the oral LD50 in the first case is estimated from a range which is between 300 and 2000. So we will use 500. But in the case of cyclohexanone, the second component, we actually have a true experimental LD50. So you will do your calculation and you will determine that the LD50 via oral route is 6,896. Now this is a case, the last case is a more complicated case. for a number of reasons. First, the first reason is that there are 4 components, but the calculations goes in the same way. So for each of the components, you will calculate a ratio, and then you will sum, and then you will normalize for 100%, and you will have your estimated LC50. There are cases where some of the components may be vapors, gas, or aerosols, so you will need to take this into account when you calculate your final LC50, considering what is the physical state of the final mixture. So if the exposure will occur as a gas, vapor, or aerosol. Now, end of the first part, and now I will show a second part which is related to the case example of the agrochemical formulation. In this specific sector, we are required to perform toxicity tests on the final product before we obtain the legal right to sell it. And the main aim here is to predict classification labeling and also personal protection equipment, first aid, and transport indications. The final classification in our sector may limit the registrability of a formulation. So it's a very important point. And at the moment, if we take into account the global landscape, we are required essentially to perform a 6-pack of acute studies for each formulation. So this means a lot of animals, approximately 60 animals used for each formulation, including an acute inhalation toxicity study. If we are lucky, we only use 10 animals. If we use the OECD 403 or 6 animals, if we use the OECD 436. But we may need to go into a full test sometimes if the LC50 needs to be estimated. So we started thinking through alternatives, and one of the easy way we found is, we have this calculation method which is included in the GHS, should be usable in all countries implementing GHS for agrochemical formulation, is usable in Europe, New Zealand, and Australia. Shall we go through an exercise and see if this method is predictive of the acute inhalation toxicity that we have found in vivo with in vivo study on existing formulations. So we went through a retrospective analysis of 225 formulations. And this is the split of in the table. You can see how the formulations were split in terms of herbicides, insecticides, fungicides, and other types of agricultural products, but also in terms of formulation types. We have liquids, solids, gels, and within the liquids we have solvent-based or more water-based formulations, so a variety really of formulations. And we compared the existing database of in vivo study against calculations made with the GHS additivity formula. The first step was to analyze the database and understand what was in there. I will focus now on inhalation specifically. So out of 225 formulation, we had studies for 123. We were able to waive most of the other studies, and so most of them were performed on aerosols and only one on a vapor formulation for a fumigant. Now, as you can see, we calculated the classification for each of the classification system that you see on this slide: GHS, Globally Harmonized System, Classification and Labeling of Products, the EU Directive, the EPA criteria for agrochemical formulations classification, and the ANVISA previous criteria for agrochemical formulation classification. As you can see, most of the cases, 92 to 96%, depending on the classification system, essentially are formulation with low toxicity and are not non-classified in the GHS CLP type of classification system, or are in the lower category, category 4, for the EPA and ANVISA system. And the categories are detailed, the comparative categories are detailed on the bottom of the slide. One of the challenges is that not all the categories have the same cutoff criteria. So, for example, also in terms of maximum concentration to test, we need to test up to 5 milligram per liter of air, if possible, if achievable, for the GHS and CLP, but we may be able to test up to 2 milligram per liter of air for the EPA and ANVISA system. Now, in this slide, you can find a comparison of the LD50 and LC50 in terms of classification category. If we take the graph on the left, we can concentrate on the GHS CLP, while the graph on the right is for the EPA and ANVISA system. So if we take the GHS system, we have 5 categories determined with animal data, and then we have 5 categories determined with the ATE calculation, the GHS additivity formula calculation. You can see the correspondences in the diagonal, and the overall number of matches is very high, is 94.3%. A similar result is obtained also for the EPA and ANVISA system. Now we have a number of underestimation and overestimation, and which is obviously expected, and this is about 5.5% for the GHS and CLP classification in total, and approximately 3.2% for the EPA and ANVISA system. So this really goes back to the evidence already found in the 60s that a 5% additivity should be assumed, should be assumed really, unless proven to be different. Now, this is just the first overview, but we did some calculation here. So let's go back, sorry, to this slide. So we obviously were worried about the number of formulations that we have analyzed, because in category 1, 2, and 3, and 4 as well, we only have 9 formulations, right? So 9 mixtures, while most of them are not classified. And we know that the additivity is particularly, from the evidence in the literature, the additivity, the theory of additivity is particularly good for high doses, but it may not be accurate for lower doses. So we decided to look at the data also in another way, and think to the question in 2 ways. One approach is using the GHS additivity formula to predict all the categories. But some regulatory agencies may not be confident enough in doing that. So we thought, okay, can we use the GHS additivity criteria to determine if a negative is a true negative, and we only trigger testing for substances with low toxicity, so with high toxicity, with low LC50, and where we need to identify the correct category in order to determine the correct personal protection equipment to be used. So 2 different approaches. One is using the GHS as it is and across all the categories. The other is using the GHS as a prioritization tool. So we use, to determine this, we went through the exercise of grouping the formulation in negatives and positives. And to do that, we decided to use the in vivo data and use the commonly found threshold in different classification systems. The acute inhalation is at the bottom of the slide here. So as you can see, the classification cutoff for GHS and CLP is 5.0 milligram per liter of air. For EPA and ANVISA category 4 is 2.0 milligram per liter of air. The accuracy is very high. The specificity is very, very high. And this is due to the fact that the true positive number that you can see in this column, hope you can see my slide. So TPFN is true positive, false negative. So the number of true negative, the number of true negative is very high. So it's 113 out of 123, and this drives the negative prediction value to be very high. So this tool is extremely valuable and extremely good in predicting negatives, and really should be used. Now, we also tried to find more information from the database, because once you go through the retrospective analysis, it's interesting to see how, for example, inhalation toxicity relates to oral. There were evidences in the literature that dermal toxicity is always lower than the oral toxicity for mixtures as well as for single components. So we wanted to verify if this was the case also for inhalation versus oral. This was confirmed. If we focus on the last line, last row of the table, you can see that all formulations that have an oral LD50 higher than 2000, and so they are not classified in the GHS. These are 99, so most of them, 96, are also non-classified for acute inhalation. So the LC50 is higher than 5.0, and only 3 of them are classified in category 4. So this is another information that could be used in a weight of evidence. So if you already have an oral test, you can use that to see what is the likelihood of a negative, and then you can do your calculation, and if also the calculation demonstrates a negative, you have 2 pieces of evidence to give strength to the fact that an in vivo test is not necessary. Now, this is just to give an idea of what is the regulatory requirement and what is the true life exposure scenario. So in our database, the LC50 was below these thresholds in a very small percentages of cases. And this is based on experiment in vivo performed for a 4-hour continuous exposure in the rat. Now, if we expose humans for 4 hours continuously, assuming a certain breathing rate, we will be exposed to 5 cubic meters of air. And so if we have concentration of 1, 2, or 5 milligrams of our mixture per liter of air, that would result in these very high exposures of 1,000, 2,000, or 5,000 milligrams per cubic meter of air. Now, these values correspond to the concentration of a fraction PM10 of dust particles expected respectively in a normal, strong or severe dust storm. So, and there are also 2 lower levels to this in this publication that you can see here. So now this is already 1 milligram per liter corresponding to 1,000 milligram of mixture per cubic meter of air is a very worst case scenario. And we have to consider that for agrochemical formulation, the exposure to the concentrate formulation is extremely limited in time, because it's really limited to opening the container and pouring into the tank for dilution and application in the field, for example. This is the example for a liquid formulation. this operation, this mixing and loading procedure is most likely to occur in open space and potentially using closed transfer systems that are now available. So another conclusion we propose is that when testing is justified, a concentration limit of 1.0 milligram per liter of air represents really a worst-case scenario for agrochemical formulation. We shouldn't test above that, and this should be taken into account when we describe our exposure scenario, especially in those geographies where exposure scenarios can trigger the need for an in vivo test. Now, based on this evidence, retrospective analysis of the database, retrospective analysis of the classification results versus obtained with the in vivo studies versus the results obtained with the GHS additivity formula, we have proposed an approach, and we are publishing this approach, and we are discussing with some regulatory agencies for policy changes potentially in the future. So specifically for agrochemical formulations and for the inhalation toxicity, acute inhalation toxicity requirement, we would like ideally all the agencies to evaluate the need for another characterization first, considering waiving criteria that are also laid out in the OECD series on testing and assessment published, that's going to be published this year for acute waiving, acute testing waiving. Consider the oral toxicity information, if available, as a part of the weight of evidence. If there is the need for another characterization, we propose to start with the GHS additivity formula and to accept negative results if they are higher than 1.0 milligram per liter of air, assuming that no PPE is required according to what is currently the policy of EPA. And if the LC50 is below 1.0, which is expected in only 2% of cases, then to reconsider the weight of evidence and the physicochemical characteristics to understand if we can assign respiratory protection without performing an in vivo test. And only as a last resort, the in vivo test should be performed, only really if strictly necessary to determine correct PPE to be used. So essentially in these sectors, I believe that if we follow this approach, we would probably reduce 95% or more of the animal testing for inhalation toxicity. So in terms of implementation, these are just some of the points we are doing. We are using it internally, but we are using this approach, the GHS calculation approach, also for all EU business cases as a predictive tool before any in vivo study is performed, also to determine if we need to start to a lower dose level, avoid lethality, avoid severe toxicity. And then we are working on improving this method and improving the acceptance of this method globally with a publication strategy, but also dissemination to EPA and other agencies, presentation to Congress, and participation in some of the OECD, but also ECHA working groups. And we are obviously working with our allies, animal welfare associations, for the implementation in global developing regulation. I would like to thank all the people that were involved in this project that are working in Dow AgroSciences, DAS, but also in the Dow Chemicals in our corporate lab in Midland, Michigan. Thanks for your attention.
Okay, thanks very much, Marco. As Amy mentioned, my name is Dave Allen. I'm the PI for the NITUDIM support contract. At this point, we'll open up for questions on the chat box. I do have one that came in. But thanks very much, Marco. A very compelling case for, at a minimum, weeding out or screening out those mixtures that may be considered non-toxic. So one of the questions that has come through is relevant directly to slide number 5, Marco, where you mentioned applicability of additivity to transport regulations, and they just asked for maybe if you could expand on that a bit, because the questioner said, I thought this was not very much the case. So the applicability of additivity to transport regulations and how well received they are, I suppose.
Well, we have used these. To be fair, there are only a few points. So the GHS has two approaches. One is the additivity, both based on the theory of additivity. One is an additivity formula, which is for acute systemic oral thermal inhalation toxicity, and the other one is a concentration threshold method for acute skin irritation, eye irritation, and skin sensitization. So what impact on the transport is the severe systemic toxicity that can be calculated with the theory of additivity, as well as corrosivity that can be calculated with the concentration threshold. And in the UN transport regulation, which I believe is called Orange Book, and I can provide references after the meeting with the chapters and where this is described, you can find this approach. And if you compare the approach with the GHS, it's essentially the same type of approach. It's based on theory of additivity. So this approach is usable because it's the UN Orange Book is also, the approaches in the UN Orange Book are also implemented in regional regulations, also in the maritime regulation, in the IATA, which is the aerial transport regulation. So in all these downstream regulations, these principles are brought forward. So if I can provide probably to the person who asked more detail after me, I would be happy to do so.
Okay, great, Marco. I can put you in touch with the questioner. The next question is relevant to the units used in the formula, I suppose. Are you always to use the concentration in milligrams, or do you ever convert it to molar concentration?
What do you mean for the calculation?
Unfortunately, the question, I'm presuming it's the actual value that is expressed. But unfortunately, the questioner just said, are you always to use concentration in milligrams, or do you convert to a molar concentration? Yes, for the calculation in your formula.
Yes, so this is from GHS, well, CLP GHS. As you see, concentration are in milligram per liter of air, or ppm for gases. So that hopefully clarifies what units should be used for the calculation.
Right.
And this is also the information you will find on the databases, because the MSDS will contain the information in terms of milligram per liter of air. Rarely, so the regulatory approach is based on milligrams rather than concentration, apart from the gases.
Okay, thank you. How do you assess formulant chemicals that are not very toxic on their own, but might be known to increase exposure time, for example, thickeners, or increase absorption, something like DMSO? Might these formulants multiply rather than add to toxicity?
Yeah, that's a good question. So those are those cases where you have evidence for one of your coformulants that they may increase, absorption or other biological phenomenon that can affect the toxicity. What I can tell you is that, for example, we had this question for agrochemical formulations, because for some formulation which are solvent-based, we often have surfactants that are used to hide the suspension of the active substances, and as known, surfactants may increase, may change the absorption profile of substances, at least through the skin, and potentially also via inhalation. So we were kind of worried about that, but I didn't show here, but the predictivity was good, was as good for water-based liquid formulations as well as for, they were comparable, as well as solvent-based formulation, including surfactants. So we excluded that surfactants may, well, at least those that were in our database may increase, absorption. Then there might be other that need specific addressing this specific question. But once you have evidence that these surfactants do not, increase, it depends really on the approach you want to take. You may be very conservative and then decide that maybe in this case you should do some more experiments and possibly an in vivo test. But if possible, we would suggest to start with the overall weight of evidence, the acute oral toxicity, if available, even the neutral red uptake. Assay, which is it's not directly related to inhalation, but there is a good relationship between non-toxicity in the neutral red uptake assay and lack of acute oral toxicity, and this was demonstrated by JRC recently. So again, also that test could be used to indicate that there is low potential toxicity in biological systems. So if you take together all the weight of evidence, probably you will be able to build a sufficiently strong case.
Okay, thanks, Marco. The next question relates to slide number 8, the next slide on your presentation. You mentioned that the form of the final formulation should be considered for LC50 estimations. Can you use data from individual components in one form to estimate LC50 of the final formulation in another form? For example, a vapor versus an aerosol.
Yeah, that's exactly the case that is here, because in the last line, so the first, second, and fourth component are aerosols, and the data that we have, the estimated ATER for aerosols forms, but cyclohexanone is a vapor. So in this case, we took a pragmatic approach, and we decided to use the lower value. You could convert. We have discussed a lot of what was the best case to do. So you can assume that, for example, cyclohexanone is category 3, if I'm correct, and then you can convert the category 3 vapor to the category 3 aerosol, so that you have a consistent aerosol mixture, and use an acute estimate for that. Or you can use just simply the lowest LC50 values, whatever is the vapor or the aerosol, and then use that in your calculation. Now, in this case, there is the worst case, the numerical worst case. You have to keep in mind that the scales are quite different. So there are cases, and I show this on here. So as you see here, the scales for gases and vapors are quite different from aerosols. So you may have cases where this is not so easy, straightforward, and you may not apply this method.
Okay, great. Yeah, thank you, Marco. The next question is really a question for clarification. When using the formula, you're only to use those ingredients that fall within GHS categories 1 through 4, correct? In other words, anything that's unclassified is not included in the formula.
Yes, exactly. Yes, right. that will add up to, so in this case, for example, second line, you will have 94 point something percent, that 95 something percent that is non-classified, so you will not take that into account. Okay, great. And the final question that was submitted is just a specific reference to the OECD, the 2016 OECD document regarding study waivers.
Do you have that specific reference?
Yes, so I think this document was this document has been voted at the April WINT meeting, OECD meeting. So that's going to be published in July. The final version is going to be published in July, and the draft version is available on the OECD website. I think in the version before the last commenting. But I can give the reference after the meeting, if that's okay.
Yeah, no, that's great. Thank you, Marco.
Yeah. It's a draft series. It's a draft document of a series on testing and assessment of the OECD program.
Right. Okay. Well, one final question that I had, since you have slide 16 up there, you have your last resort approach using a reduced in vivo approach. Have you, or are you aware of anyone that's pursuing consideration of how in vitro models might fit into this scheme to inform, additional information prior to or replacing the utility or the need for the in vivo test?
Well, we haven't considered the in vitro test in this approach because the acceptability, well, we went kind of on regulatory accepted approaches, at least borrowing them from other sectors or where available, like the OECD one or the GHS one. So we didn't integrate proactively in vitro tests here, but it's definitely something that may be worth considering, especially between step 2 and 3 for mixtures to confirm, a lack of toxicity of your mixture.
Great. Okay. Well, thank you very much, Marco, for the really nice and comprehensive presentation and responded to a large number of questions. That was really, really great. So with that, we'll close the call. I just want to remind everyone to please join us again on July the 12th at 11 a.m. U.S. Eastern Daylight Time, where we will have, the topic will be adverse outcome pathways, and we're excited to hear from Mathieu Vinken from the Free University of Brussels and Barbara Buckley from the United States Environmental Protection Agency. With that, we'll sign off, and thank you again.
Thank you very much. Thank you, everyone.